use chemically defined media or you use peptone, you use other things. They all work, but they all work differently. And you go into the fermenter, the scale fermenters, the way that it's designed and process control will be different. And then purification, obviously, you could have different columns organized in different sequences. And formulation, definitely, you could have also have different formulations. And we believe, and as many of the colleagues here would agree, that the different process means different product. So different process, if you have different processes, the product will be different. You can have the same seed of orange. If you plant it in Shanghai, it's going to grow differently as you plant it in Beijing. So different processes with the same DNA would result in different products. We took a product that Dr. Reddy produced that they claim is biosimilar to our Methera. It's called Reditox. When we just did a few analysis, I'm sharing one with you here, which is the communication exchange using chromatography. And they show significant difference between the two products. And this is only one of the many pieces of data we have. So the clinical data of Reditox amounts to 17 patient data. So what we would assert is that Reditox is not a biosimilar by our definition. A biosimilar is a product that demonstrates similarity using a high standard with the state-of-the-art analytics to demonstrate it's highly similar. So what Reditox really is an intended copy. It's just intended copy. It doesn't really meet the test of true biosimilar using the WHO definition of biosimilar approval pathway or the EMA's European Medicine Agency's approval pathway. So our perspective on biosimilars is that myself and my company believe that there should be a robust approval pathway. So biosimilars should have a way to be approved, just like generic chemicals. So the innovators, when the patent expires, my perspective is that we should enable the government and the agents should enable biosimilar producers to produce biosimilars through a robust set of standards. And from the innovator's perspective, if you look at the healthcare budget as a single pie, whether that's 10 billion for some countries and 100 billion or even trillion for some countries, that pie is not going to continuously grow. So if you look at the amount of money every country is willing to pay for the pharmaceuticals, you could say that it's pretty much going to be constant from this point on, adjusted for inflation, meaning that the growth is probably going to be 2 to 3%, as we've seen in the United States, 3.5%. So if we do not have a pathway for generics or biosimilars, then the government would not be able to afford any new products. Because that's a constant piece of pie. So if we have a $10 billion a year product that does not go off patent ever, we have a problem. So that product has to become generic to make room for the next product we bring in. So if you think of it that way, then it makes sense to keep the company going, all of us going with new products, when there's a pathway to safely for the patients to become generic. So we believe that it should have approval. But patient safety is the most important one. It must be top priority by all of us, by the agencies, regulatory agencies, and the producers, as well as the innovator, because we still care when somebody tries to copy our product, and if that product has a safety signal, we need to know about it. We will continue to work with the agencies and the industry to make sure that appropriate pathways are provided, and we will continue to innovate. So our strategy is to continue to innovate. So here's just a few examples. For anti-CD20, in addition to Methera or Rituxan, as it's branded in the United States, we are developing subcutaneous Methera, and then we have new molecule GA101 that's coming up. That's a second generation humanized anti-CD20. Anti-HER2, Herceptin, We have subcutaneous Herceptin that we're treating just like new products. And then we have TDM1, which I'll talk about with the next slide. TDM1 is the first antibody drug conjugation product that Genentech Roche has developed. And we also have produced one, so new generation of HER2. Avastin, a very popular area for all of us. We will have new ones coming in as well. And then Actemra, which is rheumatoid arthritis, we have subcutaneous formulation coming up very soon. So I will use this one to highlight what we would do next as an innovator. So we launched the Herceptin over a decade ago. This is one drug that had demonstrated overall survival rate, significant overall survival rate increase in HER2 positive breast cancer patients. It saved lives. The next generation of the same product will use HER2 as the guided vehicle and attach to that the toxin, the chemotherapy toxin, and that's the warhead of the missile. And this product is very exciting. We plan, if everything goes well, in 2012, we expect to file this product. So in the future, this is a new technology platform that Roche Genentech intend to use. That is to take the large molecules, monoclonal antibodies, and then attach that, link that to a small molecule for various unmet medical needs. So the outlook is that, that's my last slide, is that the regulatory environment for biotech industry is evolving, it's changing. Biologics are indeed very complex, and we believe that MABs are even more complex than anything else. We believe there should be a path of biosimilars, but we will continue, Roche and Genentech will continue the path of innovation-based product. Thank you very much.